Welcome to the second session of the research track on the first day of the MDF 2020 conference. My name is Kay Hayes and I'm the Colorado Support Group Facilitator. Today it's my pleasure to introduce you to Dr. Eric Wang. I first heard Dr. Wang speak, I think it was a 2012 conference, and was so grateful he was going to pursue research that would benefit our community. He will be discussing DM treatment progress, something we all want to know more about. An MDF fellow in 2012 and 13, Dr. Wang has always been interested in understanding how biological systems function, but in part entered the field and joined the team at the University of Florida because he has family members affected by myotonic dystrophy. He's personally motivated to better understand DM and related diseases and to find treatments. He's a brainiac. Running his own research lab at the University of Florida as assistant professor in the Department of Molecular Genetics and Microbiology, his research is focused on three main areas: studying the pathogenesis of microsatellite repeat diseases, in particular myotonic dystrophy, studying the basics of how RNA is processed and localized in cells and tissues, and combining insights made in both of those areas to develop treatments for people with these diseases. So Dr. Wang, your turn.
Thanks so much, Kay. Before I go ahead and share my screen, thanks for that wonderful introduction. I just wanted to, say a little bit more about myself and orient people who, maybe I haven't met in person. It's really a pleasure and an honor to be able to communicate with you about these topics today and to hopefully provide a little bit of information that's going to help make, going through this journey, together a little bit easier. I decided to get into myotonic dystrophy research, as Kay mentioned, because I myself have family members that are affected by this disease. And in college, I thought I wanted to become a doctor because I wanted to help people dealing with different medical issues. But I realized that there's a lot we didn't understand about this disease and how it really works. And so that's what really made me motivated to pursue a research career. And I was really fortunate to learn meet a number of individuals that had been doing research in this area, followed their papers. One of those individuals was Maury Swanson, who's now my neighbor at the University of Florida, also Charles Thornton, and also Andy Berglund, who really encouraged me to pursue a research career in this field, and were very encouraging that I could do it, and so decided to pursue research in this area as a graduate student at MIT, and then was fortunate to be able to gain a lot of research support to start my own lab and move here to University of Florida. And it's been exciting to watch the Myotonic Dystrophy Foundation grow. I feel like we've been really important partners together in this journey throughout the years, and I'm really excited to tell you more about the progress that's been made. And so I'll go ahead and share my screen and start this presentation. So, I was tasked with really describing this topic of treatment and a cure. Where are we now? And I think it's really important, in particular for this community, and based on conversations that have been that have been ongoing over the past few years, to really think about, what do we mean by a treatment and a cure? What is it that we're really talking about? And when you step back, I think it's important to think about, what would a treatment and a cure really mean? And I think what we're talking about is, any sort of intervention that could improve the quality of life for patients and, as a result, their families. And there's different axes through which we can think about this. So, for example, different treatments and cures might last different amounts of time. So how long would an intervention last? Would it last, just for a few days, a few months, forever? And how well will it work? What kind of degree of relief will it give you, for example? Will it alleviate all your symptoms completely, or will it help a little bit? as everybody knows, myotonic dystrophy affects many different tissues and body systems. And so different treatments and cures might actually affect, just the muscle or just the GI or just the CNS, or multiple of those tissues and body systems. And also, I think that people have been talking more and more about whether you address symptoms versus the root cause and whether one is better or worse than the other. And so I think it's just really important to address that, it's commonly perceived that, for example, a treatment might not be as good as a cure, or that they're actually different. But I think an important point for our community to remember is that this really exists along a spectrum. Again, if we just go through those different axes, in terms of time or duration, if you had a treatment that was temporary, but you could, take it repeatedly, would it essentially feel as though it's a cure, it's forever? And again, the degree of relief, some treatments are going to work better than others potentially. But how good does it have to be to make a meaningful difference in your life? I think something that's been focused on more and more now is the multisystemic aspect of this disease. So some treatments may address some body systems and tissues, but some may address more. And I think that many in our community would agree that, we don't need something on day one that affects all the systems. I think something that affects some tissues would still be really valuable and still be really important to our community. Again, root cause versus symptoms. There's a lot of discussion about attacking the RNA or attacking the DNA and fixing it or changing it in some way. But a lot of effective approaches could be simply to address symptoms, and for all intents and purposes, we may not even realize whether the intervention is affecting the root cause or just addressing the symptoms. And so I think I wanted to frame this discussion around that. Again, treatments or cures, they're interventions that may improve quality of life for patients and their families. And it's important to keep in mind that obviously there's a lot of people working on this. There's a lot of efforts being put in, a lot of time, a lot of energy, a lot of money. There may or may not be a single or complete cure, right? But there may be one or more impactful treatments. And I think that's very encouraging. But I think it's important to shift the discussion rather to just solely be focused on something that's, quote, a cure, that treatments can still be really important, and having multiple options is a good thing. And I just wanted to address that because I know this is an ongoing conversation in our community. And I think it's important that we all sort of get on the same page and really get granular about what we mean when we're thinking about this topic. So in the talk, mainly, I'm going to talk about general approaches and the overall progress. I won't really focus on specific companies or efforts, and there's a number of reasons. I'm not going to focus on specific companies. One is just that multiple companies have interactive booths at this meeting. They're in the best position to talk about their particular approaches. And there's a lot of reasons sometimes why some companies will be more or less open about what's ongoing at the company. And not all those reasons are, not all those reasons are good or bad. There's a lot of different reasons why a company might want to disclose more or less. I will say that our space to develop medicines for our community is very, very competitive right now. And that's a really good thing. There is a lot of competition to come out with treatments for our patient population. And that is reflected in, for example, sometimes when companies might be more or less secretive about what they're doing. So again, I'm going to focus on general approaches, and I'll mention some companies that are taking different approaches. I won't be able to cover all the approaches taken by all the companies, again, just because some of them aren't publicly disclosed or known. And many of you probably have attended Andy Berglund's talk earlier today, where he discussed different molecular mechanisms that underlie this disease. And I just wanted to review that just so it's fresh in our mind, and also for people who weren't able to join Andy's talk. He did cover this idea that you might have short CTG or ccTG DNA in unaffected individuals, and then expanded repeat DNA in DM And he used this analogy of different height trees or different size trees. So again, short CTG DNA, sort of you can think of as a lot of short trees in maybe an orchard. And then in the context of expanded repeats, you have trees that grow at different rates and some get really, really big. And so what I'm going to do is extend this analogy further. And Andy and I did coordinate on this so that we could have a unified analogy. Maybe next time we'll have a different analogy, but this year we chose trees. And I'm going to extend it further to describe this cell. You know, every cell of your body we might think of as a sawmill. And you've got a lot of RNA in the cell that needs to be processed. And one of those processors, those proteins that is around to process it, is the muscle-blind protein. And you can think of this as basically the mill workers that are moving around inside the sawmill processing these logs. And in myotonic dystrophy, you have these expanded CUG RNAs. You can think of that as a really large log that pulls a lot of mill workers from their other smaller size logs. It takes up a lot of time. It gets them stuck there. You know, similar to how the RNA basically distracts or sequesters the muscle-blind proteins. And as a result, you know, those mill workers, the muscle-blind protein, it can't do its normal job because it's stuck on those large logs. And so just to sort of back up and review again the molecular pathology in myotonic dystrophy, you know, we know that these repeats are expanding rapidly throughout the lifetime of individuals. That can be thought of as, you know, trees growing. So you go from small trees to huge trees, or short CTG or CCTG repeats to longer ones. And that gets made transcribed into RNA in the cell. And you can think of that as essentially the trees are coming into the sawmill and they go in, they're inside now, and you have to mill them. The long RNA ends up trapping the muscle-blind proteins, and the logs do the same thing. They trap the mill workers. As a result, muscle-blind can't do its normal job and process other RNAs, and you get downstream cellular pathology. And in the sawmill, you know, all those other logs go unmilled, and you can't fulfill those orders. So you have a problem with your customers. So just with that review in terms of the molecular mechanisms, you know, where are we now? So first of all, I think it's important to acknowledge the work of a lot of different scientists over many years has led to that, you know, seemingly simple diagram, that very sort of linear flow chart that I showed. You know, it took a lot of work to get to that point. And of course, the way I'm presenting it is certainly simplified. There are other things that happen in the disease that I'm not showing here, but really I'm focusing on a major pathway that we think is important. But, you know, it took a long time to get to that understanding. And it might never be complete, but what's really fortunate and amazing right now at this time is that it seems as though we have enough understanding to really attempt to intervene in a sensible and potentially productive way. So because we understand the pathways that are causing disease, there might be ways to go in and make the changes you'd like to make in order to, you know, improve the biology essentially. And again, really it's the knowledge of the effective biological pathways, the knowledge of what's happening in cells. It provides ways in which we're able to think rationally, how would you intervene? And so we go back to that diagram, you know, every single step in this process, in this pathway, the expansion of the repeats from something short to something long, the copying of the DNA into RNA with expanded repeats, the interaction and trapping of muscle-blind onto the RNA, and even some of the downstream changes that happen as a result of this, or as a result of just the RNA having expanded repeats. You know, we can understand a lot of these and potentially block these processes. And so what I want to do is go through each of these arrows and describe what is being done in the medical research, biotech, and pharma fields to try to address each of these processes. And so the first part of the pathway I want to tackle is, repeats are unstable and they are expanding. So a logical way to prevent them from expanding, which would be, very early in the sort of the biological pathway, is to prevent the expansion. And so you're preventing these trees from growing up. So there are, in fact, approaches that are being taken. There are companies that are focused on affecting or modulating the pathways that are responsible for repeat expansion. So triplet therapeutics is one of those. And then there's even parties that are out there, companies that are working on not even just stopping the expansion, but potentially removing that bit of DNA so it can't expand, so it's not there anymore. And CRISPR Therapeutics and Vertex are attempting to do this. Now, that would be fantastic if you can do that. But a lot more work has been done, I would say, in the context of the next part of the pathway, where if you do have those expanded repeats and they're made into RNA, how do you solve that problem? How do you block that process? So in using the analogy, you have big trees, but just don't bring them into the mill, or somehow destroy them very quickly so that you're not distracting those mill workers. Analogously, in the cell, you really would want to prevent production of the RNA. So don't even let it get made in the first place, or you might want to destroy it, pretty quickly after it's made. And here, as you can see below, there's a lot of different companies that are working on that. So Ionis and Biogen have been some of the pioneers in this area. And there have been other parties that have come along. Dyne and Avidity have exciting programs ongoing, and I believe they have talks as well in this in other parts of the meeting. Audentis has pursued a program where they just try to destroy the RNA, and so has Lacana. And then Syros is actively working on ways to prevent the production of the RNA. And so there's a lot of excitement around this area, and potential proof of concept that's been achieved in other diseases. So let's say that you got the RNA and it's floating around, the logs are in the mill, and you still have to deal with it. What are strategies in which you can prevent potentially trapping of muscle-blind proteins or the trapping of the mill workers? There's a variety of ways you can do that too. So one would be to basically tell the mill workers, you don't have to deal with this log, just leave it. Go away and deal with your normal job. And that's something that's being pursued by companies such as Expansion Therapeutics, which are developing small molecules to prevent muscle-blind from binding to the RNA. And other parties such as Nubase, I believe, are doing similar work with peptide nucleic acids. And then the other strategy would be, okay, you've got a lot of mill workers stuck on the logs. Well, just hire more workers, right? Bring more into the sawmill, and that would be equivalent to adding more muscle-blind to the cell. And there are parties trying to do that too. So Arthex Biotech has found a way to try to upregulate or increase the amount of muscle-blind in cells. And then finally, we are getting farther and farther from, quote, the root cause of the disease. But I want to emphasize that, a lot of the treatments that we have in medicine today, they do in fact address symptoms, and they're really powerful and they're really effective. And so I don't think we should discount those. I think there's a lot of valuable approaches there too, which is to figure out what are these changes downstream of repeats or trapping of muscle blind? What are these changes that occur? And are there ways to affect downstream biological pathways to really address symptoms? So, there are a number of companies that are working on methods to do that and developing compounds to do that. So Lupin Neurosciences, in fact, is responsible for, they make mexillatine, which many of the individuals in our community use and take and find valuable. And then there are other parties like AMO Pharma going after a pathway called the GSK3 beta pathway, and they have an ongoing trial. Harmony Biosciences is going after trying to address sleep issues in the disease. And so I think these, and there may be others that I'm missing here, are also really valuable approaches that find a way that even though you have a big log in the sawmill, you're able to fill your mill orders somehow, right? So I wanted to outline all of these approaches because in the end, regardless of what the particular intervention is, what really matters is, does the medicine work well enough to improve symptoms? And of course, that's going to be, that might be different for different people. So some people might find that something that addresses the muscle symptoms is, completely, and such that they can improve in muscle strength, for example. Maybe that's really important to them. Maybe others find that stopping progression is good enough. Maybe others want their, sleep issues to be addressed, or, cognition to be addressed. Whether, whatever that is, I think that's what we need to be focused on. And then, of course, we need to weigh, any sort of ways that the medicine might modulate biology. We have to weigh that with safety. It has to be safe enough to be acceptable for our population. And so I think that these are really the bottom line in terms of providing medicines, and these are the criteria by which, for example, FDA will evaluate any potential medicines. I wanted to take some time to discuss, some of the key challenges that are out there right now to develop medicines for DM. There are a lot of approaches that can be taken to treat DM cells in a dish or in mouse models. Andy described a number of mouse models that are really important because they're really one of the best ways to study how a compound or how an approach works in a living organism that has molecular and phenotypic symptoms of DM. It's really one of the best ways to evaluate how things work. But as we all know, mice are not humans, and translation to human is quite difficult. And I want to highlight one of the difficulties, one of the challenges, and I think a challenge that has motivated a number of players in the space now. So a huge barrier to some of the therapeutics right now is effective delivery to the tissues that matter. And what do I mean by delivery? I'm going to go back to the sawmill analogy in a bit. But what I will highlight is one example of a previously tested therapeutic developed by Ionis and Biogen. And Andy may have hinted and mentioned this earlier, but basically what this therapeutic does is if you have your toxic RNA here and you have muscleblind stuck to it, something called an antisense oligonucleotide is designed so that it can actually recognize the RNA sequence and make a nick. It's actually able to recruit machinery in the cell to cut the toxic RNA sequence. And because of that nick, that RNA can naturally get degraded, and the muscle-blind protein can go off and do its thing. And so it's really similar, again, to going back to the log analogy. You've got these huge logs. The antisense oligo, it's like this crazy four-headed chainsaw, and it can come along and it can convert that to sawdust, like very, very efficiently. And that's one way to think about the antisense oligo. Okay, so that's very exciting, and potentially very effective. But going back to the sawmill analogy, our sawmill is in a cold place. It's indoors, everything happens inside. And it's really difficult. That four-headed chainsaw, it's just too big. You can't get it into the sawmill. You can't get it into the windows, the doors. You really need a key. You got to open up these big barn doors to get the chainsaw in. It just won't get in very well. And so this was the conclusion, one of the conclusions that Ionis and Biogen reached was that, yeah, if you can get the chainsaw in, it works pretty well, but it's challenging to get it in. And so multiple efforts now are focused on really finding keys to enter the sawmill. And I won't go into too much detail on methods to do that, but there are other presentations in the meeting that will describe that further. And other parties, other efforts are focused on really finding smaller medicines that can easily enter. So what if instead of a chainsaw, you had a stick of dynamite that could recognize that large log? So it knows exactly to go to that really big log, and you can toss that in the window. You know, that might fit. There are a lot of efforts focused on doing things like that as well. And so I really wanted to just highlight this example because it illustrates sort of practically what the challenges are. We know what's happening to a large extent, what's happening inside the cell, but now we have to find ways to be able to modulate that. All our bodies have really extensive mechanisms to keep foreign stuff out, right? You want to keep the outside world out, you want to keep the inside stuff in. And so it's really difficult to get around that. And you know, not only is the DM field trying to figure that out, but I would say, you know, many disease fields are trying to figure that out. Many neuromuscular diseases, neurological diseases need to find ways to be able to get material from the outside world in to be able to make the modifications and the changes you want. You know, again, where we're at, many companies are actively working on potential medicines, and some are planning clinical trials. And I can say confidently that when I was in graduate school, you know, and when the foundation started, there were maybe 0 or 1 companies thinking about this and in the space. But now, I believe at least 20 companies have preclinical programs in myotonic dystrophy. Most of them are focused on type 1 myotonic dystrophy, but there are companies that are thinking about and planning work in myotonic dystrophy type 2 as well. And I know that that's important to our DM2 population, that it feels as though DM1 is farther along, but it's not getting forgotten. There are companies thinking about it. And of course, all the work to enable actually developing treatments and doing clinical trials in DM2 are, you know, there's a lot more attention being placed on that now than before. A handful of companies, some that have started recently, have stated they want to initiate trials within a few years. So that's very exciting. And then a few have begun let's see if I lost my screen. Yep, a few have begun or completed clinical trials as well. And a lot of this information is actually available on the Myotonic Dystrophy Foundation website. And I wanted to address, you know, why has the amount of interest gone up in developing medicines for DM in the recent years? You know, why so much interest now? I think there's a number of reasons. Really, we have a much better basic understanding. And by basic, I mean, oh, sorry, I've got a message. My screen is not showing. Let me go back. Thank you. Okay, hopefully that's better. Great. So, you know, why has there been a lot more interest in developing medicines for DM in the recent years? I think one reason is that we have a much better basic understanding of disease pathogenesis. So what do I mean by basic understanding? You know, the basic science, what's happening in the cell at a molecular level. It's really matured to the point where we think we have logical ways to intervene. I think another really important aspect is, let's say that we had a medicine that would work perfectly today. If we don't have a good way to measure how well it's working in patients, then we're never able to prove that it actually works, right? So there's a lot of work that's being done by a number of investigators, a number of clinical investigators in the field. And I would say that our field is very collaborative in trying to mobilize those efforts. There's a lot of efforts that have been done to develop ways to measure how one might respond to a medicine. So natural history studies, how does the disease progress over time? What are the most important symptoms? Can we measure the degree or severity or impact of those symptoms in a quantitative way? And ways to measure molecular aspects of the disease, biomarkers, for example, to get an early read on whether a medicine is behaving as expected. And I think that part of the reason there's also been a lot of interest is because there have been pioneers, certain drug companies that have paved the way by observing some signs that certain approaches might actually work. So getting some initial proof of concept that maybe isn't quite good enough yet, but has some hints that the approach might actually be very productive. I think a really important part of this also is that the patient voice, all of you, and the foundation, telling the world that medicines for DM are important and valuable, and in what way, and being specific and precise about that, that's hugely valuable, because that helps drug developers design the right measurement tools and right endpoints that will be eventually used to actually get these drugs approved. Just to summarize before I take some questions, in the outlook ahead, I want to, leave you with the feeling that, yes, while we're not quite there yet in terms of having a treatment, or treatments, there's a lot happening in this field right now. There's a lot of competition. There are a lot of companies involved who want to be, the first, or even the second, or the third, to be successful in providing treatments. I think that another thing to note in our patient population is that, the disease affects different people in many different ways. So what that means is that there may be a lot of space for different approaches, and there may be a lot of value in different approaches, and that it's not that one particular company is going to come along and there's going to be a magic bullet. It may require a couple different approaches, but that's actually good because it provides our community different options, and that's something to keep in mind. There's a lot of ways you can get involved and remain involved. Again, because of the natural history studies that have been done in our community, we have a better sense for how this disease behaves and how people are affected by it. Participation in those studies is really important. Because again, this disease is quite variable across different people, it is not trivial to recruit for clinical trials. So, many of you may have tried to do this in the past, tried to participate, and sometimes you might be excluded for certain reasons. There might be certain criteria that you have to satisfy. And that's not because, your participation is not valuable, for example, but as I mentioned before, it's really important to be able to accurately measure what kinds of treatment effects there might be in response to medicine. And sometimes that means finding patients with a certain profile or a certain type of phenotype, because that's going to maximize the ability to be able to detect different positive effects. But always participating in these clinical trials is really valuable and important. It really helps to accelerate things. And then continuing to let people know, letting the foundation know, letting different investigators, or even when you go and talk to the different drug companies at their booths, letting them know what issues are important to you. That's hugely important, and I don't think that there can ever be too much of that. I will say that the Myotonic Dystrophy Foundation has done a fantastic job to provide additional resources that are more detailed than I could go into in this presentation. You can find those at these links on the website, and it's continually updated and has a lot of great information. And so with that, I will be happy to take questions. Again, thanks to Myotonic Dystrophy Foundation for helping me get my first independent research grant as a postdoc, as a fellow in 2012 and 13. And I'm happy to take questions. Great. So I see in the chat that there are a number of questions. There was a comment about not to forget pacemakers and defibrillators. And that is an example of an intervention that, yeah, it's not exactly a medicine that people might think of as a traditional medicine, but it's an intervention that has a really important impact and can save people's lives, for example. The next question, is there any consensus on which symptoms are the most critical to address given the heterogeneity of the disease? I would say that I don't know if there's a consensus per se, but there have been really effective, really nice studies done by various investigators. So one study that is often cited and used as supportive information to justify developing drugs for this population is done by the Rochester group, Chad Heatwall. He's done patient-reported outcome studies. And what seems to come up on top, and this doesn't discount the other symptoms, of course, but problems with hands and arms, that may mean muscle strength. That may also mean myotonia, for example. That affects over 90% of people. And then the other aspect that is really high on the list is fatigue, and whether that's fatigue that's sleepiness or whether it's fatigue in terms of muscle fatigue. It's not clear how to separate that in the survey results, but that also affects over 90% of individuals. And then of course there are other issues that we hear about a lot, for example, GI issues, cognition, and others. But I would say that in terms of the symptoms that are most commonly reported, those are the ones. There's a question about what gene therapies am I most excited about. So gene therapy, and I should back up because different people define gene therapy differently. There's a gene therapy that can be thought of as just modulating gene expression. And so I would put the antisense oligo work into that category. I think that's very promising given the new efforts to improve delivery. I'm excited about that. And then you might be also referring more specifically to, formal gene therapy, which is using, something called an adeno-associated virus to deliver new genetic material to cells. The reason that that field is quite exciting right now because there are other companies that have pursued this for other diseases with success. So spinal muscular atrophy is one in which gene therapies are being pursued. And so, there are a number of companies that are trying to introduce material into cells to affect the biology by gene therapy. I think that field is quite exciting. But, in the end, I'm excited about really all of them. I really want to see something succeed. So I'll just leave it at that. Has COVID slowed any of this progress? I would say yes. Certainly COVID, I think, for everybody has slowed some sort of progress. But I would say that probably many academic labs have been affected by COVID because of procedures that require distancing or even just complete lab shutdowns. I do know that a number of biotech companies are affected too because they've had to deal with similar issues. But researchers are really excited to get back in the labs and they've found ways to be able to do it safely. And so COVID's been hard for everybody, I think. But a lot of labs are up and running again and sort of working at normal capacity. Are there companies focusing specifically on congenital or juvenile DM1 patients? I know that there are companies thinking about it and actively thinking about it. I don't know that I could say any that are specifically focusing on it at this time, but again, a lot of this information may not be public. What has been observed in the brain of DM1 mouse models? You know, studying the central nervous system is a growing area, and this has really been pioneered by a number of individuals. First, sort of going into human studies to do imaging studies, noticing that there's something called white matter atrophy, which has been observed by MRI and other types of imaging in human. In the brain of DM1 mouse models, my colleague Maury Swanson has found a lot of the similar mechanisms that people have observed in muscle, but the players might be different because the brain just has a different set of players than the muscle. It's sort of like you might have a sawmill that deals with walnut and another one that deals with cedar. And so you just have different settings, and the principles are largely the same, but the substrates might be slightly different. And so there is a lot of work. My lab in particular is very interested in studying the brain and looking at the CNS issues and developing new mouse models. Does my personal experience with my dad and brother impact how I do operate as a researcher and thought leader in the scientific community? It does. I think that it's, for me, I have an unusual situation of straddling different, different spaces, the academic research space, being in a family, also helping a number of biotech companies. And I think that the personal experience really helps to focus on the patients first and to make sure that the work that's being done has the greatest relevance possible to the patient community. And I think that is important, and it helps to guide the types of decisions that I make on a daily basis. Biomarkers that are indicative of a successful therapy. So there are many different biomarkers that are being developed, but I would say that some of the most commonly cited you might have heard of are these splicing biomarkers. So again, the muscle-blind proteins, Andy may have mentioned that they're involved in a cellular process called splicing. And it turns out that this is a very reliable indicator of muscle-blind function. And so the biomarkers that involve using splicing, at least in muscle, that's been where it's studied the most, are promising, I think. And Charles Thornton really has done a lot of the work here to establish that. And then there are active efforts to identify biomarkers that might be indicative of success in other tissues like the CNS, and there's a lot of efforts ongoing for that. What are some typical cognitive symptoms in DM2? I think the best way to answer this would be to point you to some literature, but just from my reading of the literature and discussing with people, I think there is fatigue, hypersomnia, executive functioning challenges, planning and organizing. Some individual to describe that something that may have taken a short amount of time to write or to do things for work might take a lot longer, and so those are similar to what you see in DM1. Are there unique aspects of DM2 that require a very different approach? And I think that there are some unique aspects that require a different approach, but then there are others that don't. And so if you think about that diagram with the pathway of, whether you have CTG or CCTG repeats, if you're going to go after, for example, destroying the RNA, well, the RNA sequence for the DM1 gene is different than the DM2 gene. The principle is the same, but this particular sequence might be different. Let's say you want to go after something that's more downstream, like the trapping of the muscle-blind protein or some downstream effect that is similar. Maybe the symptoms are similar in DM1 and DM2. In that case, you might be able to take the same modality, the same medicine forward, but then you also have to think about, okay, when you're measuring the effect, if a DM1 population has a different severity or type of experience with this symptom than the DM2 population, you have to design your clinical trial appropriately as well. So So I would think about it in terms of the modality versus the way to measure as well. Are there any stem cell therapies specific for MD? There are some academic publications that have discussed this. For example, addressing, making genetic, modifications to cells, DM cells, and then maybe transplanting them back in the body. And so there is some literature on this, but I don't know of any active drug development efforts for it. Participation in studies is dependent on location. Do DM studies lend themselves to participating remotely? I think it depends. So this is right in that participation in studies, you can't be too far away from a center. I mean, it's not as convenient. I know that for that reason, the DM Clinical Research Network, again, led by Charles Swinton, they continue to add sites to try to make it more accessible for as many people as possible. Travel and having distance to get to different places, that obviously is going to impact the ease with which you can participate. Also, you worry about, let's say you want to study some sort of symptom that affects fatigue or something, where if you go on a long trip, you're probably gonna be more tired, right? So I think that is really important, and that's something to think about. Some companies that do clinical trials do offer to pay for travel, including often it's just by car, but sometimes even by flight. May be a possibility depending on how what the trial looks like. And so I think it really depends on the particular readouts that are being made. Can the process of generating abnormal muscle cells be influenced by the environment or lifestyle? I think always. Environment and lifestyle and healthy habits can have a big impact on how one can live with the disease. I think there can be there can be an attitude where, I've got this expansion and I've got these symptoms, so I'm not even going to try to be healthy because what good is it going to do? But I do think that that's not a healthy attitude to have. I think that there is a lot one can do, eating well, exercising, obviously within the limits of what your body thinks you can do to maintain a healthy lifestyle that's going to have a positive impact on disease outcomes. There isn't a whole lot of formal research on this, but there is more and more. In fact, there have been a few recent papers looking at the impact of exercise on myotonic dystrophy patients with generally positive results. Would there be any difference between drugs with patients of DM1 on the brain versus the muscle in terms of different therapies? Yes, I think that depending on the type of medicine and whether it can get into the muscle versus the brain, there would be differences. But there are parties out there that are trying to develop approaches that would affect the muscle or the brain or both. And obviously both is fantastic, but you have to think about, let's say, if you try to go for both and aren't successful, would you rather go for one and be successful? I think these are the types of things that the companies have to think about, and hard to make that determination sometimes. Would there be any difference between drugs with patients of DM1 on the brain versus, oh yeah, sorry, I just read that one. I think Newbase believes it has the key to the sawmill and it has high precision, correct? The slide had them as a muscle line releaser, and I will defer to Newbase. I believe they have a presentation later, but I think, just to speak generally about their approach, the idea is that they might be able to essentially take the four heads of the chainsaw, take them apart, get it into the sawmill, and then put it back together inside. And I'll let Newbase sort of take that in a later session, but I think that is the concept. Do you think that defibrillators should be used prophylactically for members of a family with DM1 when the father and one of the children have used theirs multiple times? I don't think I can provide specific guidance on that, but I do think that that's something that there is literature in the, if you go to the disease handbook or the web page section that has guidelines for cardiac care. That's something to take to your cardiologist and make decisions, because risks and benefits for each individual patient need to be weighed. Is it possible for people with MD to have a remote interview with an experienced clinician to discuss any current appropriate therapies for them, or any studies that might be relevant to participate in? I don't know, but I think that with this day and age, with COVID and with everything so heavily moving online, I think it's a great idea. It may be happening already, I'm not aware of it, but it may be a really fantastic, effective way to engage more people. There's often a lengthy delay between promising data in models and a drug entering clinical trials. Can you describe the toxicity studies required before clinical trials can start, and discuss how long they typically take? Yeah, this feels like a black box for many people, right? Why do you, when you see a paper published in an academic journal, for example, and then nothing, it seems like nothing happens, or it takes a long time. It is a little bit opaque, because typically what's required to actually come up with a medicine that's going to go in people and be safe, it's a very different exercise than generating proof of concept in an academic lab to show the possibility that an approach might work. And you know, in terms of the toxicity studies, it really depends, right? And it's going to depend on the particular therapy being taken, the particular type of therapy. You can have instances where your toxicity study might need to be, a month long in rats, but for other cases, it might need to be like a 6-month study in non-human primates, for example. And it really can depend on the type of drug. And so the answer really is that it varies. It can vary, but it can take a long time. And often what you don't see publicly, but what's happening behind the scenes, is that time between something being published in a journal and, say, a clinical trial, it's not necessarily just the toxicity studies that are being done. It's also likely making improvements to the candidate, to the medicine. It's trying to improve it to make it more potent, perhaps to make delivery better. There's a lot of work that happens behind the scenes, and often it's quite hidden, because that becomes part of what a company is going to live and die by. And so there's good reasons that, for example, a party might not be able to share that publicly. They're working hard behind the scenes to try to make things work. Do small molecules penetrate the CNS? Do you anticipate treatment of DM may require combinations of treatments? So the first one, do small molecules penetrate the CNS? Some do, but some don't. And there are some guidelines that have been thought to be helpful to predict whether small molecules can get into the CNS, but they're just guidelines, and they're not always accurate. And so in the end, these are studies that scientists do objectively to actually measure the penetration. How quickly does it get in? How much gets in? How long does it stay? How does it get cleared? How quickly does it get cleared? All of these sorts of things need to be worked out, not just for small molecules, but for any medicine. Do you anticipate treatment of combinations of treatments? I think initially, yeah, I think so. I mean, I think you could imagine, for example, some people right now, at a very, many people are taking Facilitator to address myotonia, and then some people out there you might be taking Provigil. For some it has an impact, for others it doesn't, but you already are taking a combination of treatments in a way, right? But what we hope for is more potent approaches, that would help address more of the symptoms. But I think that is the direction, and we are seeing that, for example, in the spinal muscular atrophy space, that there are very, very promising approaches. One, an antisense oligonucleotide, and then a small molecule, as well as a quote gene therapy, that are all looking quite promising, some of which are approved. And there could be some different options for different patients. I don't know that anyone's taking more than one of them at a time right now, but that is the direction you might imagine things going. My thoughts on personalized gene therapy, and is there a market for this? I do think that this is a really good question for this community, again, because of how heterogeneous the disease is. It can be very different for different people. And so, I feel, I don't know if I feel qualified to answer, is there a market per se? But for example, if you could come up with gene therapies that could adjust themselves based on what someone's symptoms are, you know, I think there would be a market for that. And I think that could be the future of gene therapy, not just for DM, but for many different diseases. Do I still see people taking quinine sulfate instead of mexiletine for DM? I don't know. I actually am completely unqualified to answer that question. I think that's something Nick Johnson might be able to answer that in the next session tomorrow. Does DM1 have similar prevalence in different countries and regions of the world? It does have different prevalences in different places, but we can only answer that to the extent that it's been studied extensively enough in those places. And so there are parts of the world, like the Lake Saguenay region of Quebec, that has an extremely high incidence, perhaps 1 in 500. And the reason is because there's something called a founder effect, where there was an initial family that settled there, and then the gene pool has not expanded outward from that location, you know, over a number of years. And so there are locations with higher prevalences, and then there are locations, you know, with lower prevalences as well, it's thought. And so I see that we are coming on the end of our time here. I am happy to also answer additional questions if you want to message me through the Pathable platform. And so maybe I'll just, I'll answer this one more question and then close. So the final question is, does effective therapy targeting for different tissues of the patient require different levels of correction and rescue? I think this is I don't think we really know. So for example, if you need a certain you know, rescue in the muscle, do you need the same rescue in the CNS in the same person or not? I don't think anyone really knows the answer to that question. And so I think that, you know, natural history studies that, you know, specifically look at that, and there may be some literature that addresses that to some extent, but at a molecular level, I don't think we know the answer to that question. So ideally, having therapies that can work regardless of the severity of disease in different tissues would be really ideal. So I'll just close there and again reiterate that you know, there's a lot of work that's really exciting, and that you know, the entire community is doing a lot. The Myotonic Dystrophy Foundation is doing a lot to try to provide information to keep everybody up to date, to try to provide information to drug companies and investigators to help them focus on the issues that matter to you. And I would have loved to see all of you in person in San Diego, but we will just have to wait until next time. And so thank you for tuning in, and I will see you around virtually. Thanks a lot.